each of our sessions are recorded, and I post these recordings on our webpage. Um, and Laura McClure, a local, kind of asked about the party. So I might just begin with kind of the group photo. I'm not sure if folks have seen that, so let me see if I can find it. Yeah, there it is. So let me go back and share my screen. Welcome, David. Welcome, Scott.
Hi, Steve.
Um, so this is a photo from kind of late in the afternoon in my backyard in Dixon, New Mexico. So my fellow nonprofit partners are Miss Janice and Miss Lynn. These are these amazing high school teachers that I work with in Espanola, and a couple of our college interns, Neil and Alicia and Jade. Minna is the one who really got this whole thing started. Hefet was one of the first college mentors that I worked with. He's now full-time at Los Alamos National Laboratories. Um, Malabika's done a lot of the fashion design. Margaret is a nursing professor that we work with. Ali is a PhD student in computer science at UNM, who is really kind of leading the app development. Um, Bo is a new student of mine in engineering, who's a Iraqi war vet who also has Parkinson's at 36. He's really giving us the lived experience daily of what it means to have Parkinson's, and how we can best, build some assessment tool to turn that's not so invasive, but yet is informative enough that we can use that to tune the glove. Um, and hopefully some other folks. These are like an amazing group of people that I'm really, really lucky to be working with. And it's just kind of a snapshot of one piece of the party. Um, and so I'm hoping the group will grow at maybe our second annual launch party. Well, we're not going to relaunch something, but we will probably relaunch something, a piece of what we're up to. So um, so with that, then I might want to kind of move back into our notes, although even on that website. So let me kind of maybe go back to the journal club. So for especially for the new folks that are joining us, um, we're a pretty informal group. I mean, we're here to kind of learn together about Parkinson's. We're all students. I am trying to attract more high school and college students to join us. Um, Living in kind of an impoverished area, rural area, almost all of our students have elder duties or childcare duties, in addition to working full time while they're going to school. So I don't get a lot of their bandwidth here. I push some of these lessons on them in the after hours since the recording is there. Um, but I have yet to really win them to this kind of extracurricular kind of pretty broad tour of the brain via kind of pathways that are involved in Parkinson's. Um, but it's meant to be a learning device and a kind of and a bonding device for us to kind of learn together, for the participants to meet some of the makers and makers to meet some of the participants, and also just to share, experience with the glove. And so I don't want to be yapping more than 30 or 40 minutes each time. Um, and so hopefully what I'm yapping about will be, relevant, helping us better understand our brains. I'm an optimist. I do believe we can understand our brains. And in some sense, we must understand our brains because we're poisoning ourselves with the environment, and we need a way out, right? And that means kind of finding at least antidotes to the poison or kind of workarounds, drugs, surgery, therapies like the glove. So we began in June as a way for me to kind of better understand where people are coming from, and also to kind of meet up with my students as I was traveling for the summer. So each of these sessions are recorded. Um, for for folks that are new to our group, if you just want to go back and take a look at one of them, on the third session, it was joint with Kanwar Bhutani. So Kanwar was uh, one of the few that was, and he had an acute response to his first wearing of the glove, and he's been wearing it for 7 or 8 years, even after undergoing DBS in the past few months. So he's an amazing asset to our community, trying to understand He was diagnosed in his 30s again, so he's had Parkinson's for years. His journey is pretty powerful, and he's an amazing speaker. So to kind of go back and take a look at that third video would be a great place to start. Um, since then, we've moved, into quite a few different kind of, dimensions. And and I guess I'd really like rather talk with my hands, especially to give people some orientation for what we're doing. I mean, there are no prerequisites for this. I'm kind of hoping it's largely a kind of a visual tour of the pathways in the brain. The anatomy, though, is somehow never
Good.
quite enough. We have to really understand not just where the parts are, but how do the parts communicate. So there's some neurophysiology. All of it is motivated by kind of trying to have a deeper understanding of the glove in particular. Um, but then also, parenthetically, what are the other treatments, especially, kind of how do drugs work, and how does DBS work? And so we began kind of from the outside in. The glove is on your fingertips. And naively, a lot of people see the glove on the fingertips and it's thinking, oh, well, the glove is treating the hand. The glove is stopping tremor by treating the hand. And that's definitely not the case, right? So the glove, we believe, is rewiring the brain. So in our first couple of sessions together on brain networks, we kind of trace the signals from the fingertip to the cortex. And that's a very, very beautiful, simple, straight pathway from your fingertips to your spinal cord to your thalamus to cortex, the stuff up here. Very straight pathway. Beautiful, fast, reliable. Um, and then we started looking for, well, what happens then once the signal gets up into cortex? Um, and and it's a piece of that is even in our first picture in in the notes we're reading for today, right? Or we've been reading for the past couple of sessions. And so once the signal gets up into cortex, um, there is a path back down to the spinal cord, back down to muscles. Um, but that path is highly modulated by this kind of internal dialogue the brain has with itself, right? And so signals leave cortex and begin to enter the basal ganglia. The basal ganglia, ganglia is like a nucleus, a small grouping of cells that are more or less loyal to one task. It's never that clear in the brain that there's a lot of multitasking going on. But the basal ganglia are a kind of subcortical set of nuclei, of patches that are implicated in voluntary emotion. The high level belief is that once the cortex volunteers to execute emotion, it needs to select that motor pattern from a list of 1,000 other things you could be doing with your hands and your feet and your muscles. And the basal ganglia is then there as the filter, the selector. It kind of disinhibits the voluntary motion and inhibits the rest of the motion. All that inhibition requires dopamine. Dopamine is created in the SNC. So there'd be lots of acronyms, friends. For this is the Substantia Nigra pars compacta. This is the one of the dopamine factories in the brain. It is part of the basal ganglia that feeds the input structure of the basal ganglia, which has a big effect on what gets done in this loop. Okay. And so we've been discussing the basal ganglia for the past month or so. In particular, the putamen, how things kind of get into the basal ganglia, and then the GPI, how things kind of get out of the basal ganglia. And this loop, this gets that's the decider of what motion gets executed by your muscles. There's no indication that there's degradation of any of these signals from the brain down to muscles for Parkinson's folks. The degradation happens in here, right? So there's these inhibitory paths, and these inhibitory paths are heavily influenced by dopamine. And that's where we're going, our last meeting, and that's where we're going for a deeper dive today, is to understand what this circuit looks like with and without dopamine. It's a circuit in the basal ganglia. We'll take a look at this in some 3D brain mapping. Um, yeah, so that's a very quick review for folks that haven't been with us the past few months about what we're up to. There's a straight path from fingertip to cortex. Cortex has a lot of stuff going on. We learned last time that every cubic millimeter of cortex has 100,000 neurons and a billion
Wow.
synapses and 2 miles of cable. A cubic, so a cubic millimeter of cortex has 2 miles of cable. Cable are the things that connect the neurons to each other. There's 100,000 neurons in there, and they meet at a billion synapses. So the brain is just densely wired. And so we looked last time at a chunk of cortex, kind of color coded for us, but what's happening, at the micron level. We went deeper and deeper, and we saw synaptic vesicles crossing a synapse onto the spine of a dendrite, and then that information being passed on to its cell body where the cell fires. So that's a kind of cartoon that we watched last time. Um, and and we learned, that so this is a synapse coming from cortex into basal ganglia, and dopamine is messing with every aspect of this communication. Dopamine is modulating the release of neurotransmitter, it's modulating the uptake of neurotransmitter, and it's modulating the ion channels here that open when neurotransmitter flows. So if this is a synaptic bouton and a spine in the basal ganglia, this whole thing is swimming in dopamine. And to give an idea of that kind of what the swim might look like, um, and so uh, this is a highly informative piece of spaghetti, you guys. I mean, it's hard to kind of justify that this is not just noise. There's this 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 is an Olympian effort to trace a single axon from one of the dopamine producing cells. So, and this is most everything we know about Parkinson's, at least when it comes down to neurophysiology and biochemistry, comes from animal models. So this is the axon of a rat, right? So this is the cable coming out of a single dopamine producing cell in the Substantia Nigra pars compacta of a rat. And this thing is dropping, this thing is bathing the putamen, the input structure for the basal ganglia. It's bathing that thing in dopamine. This cable is leaving one single cell in the substantia nigra and dumping dopamine at 350,000 places. This is about half a meter of cable coming out of one individual cell. The rat has 20,000 cells. The cell is kind of nondescript. It has a few dendrites going to various places, but its axon does this crazy stuff. And so in future talks, we will get into kind of the biochemistry, um, because the dopamine that's sitting there, well, we with Parkinson's, you're not making enough dopamine. So are there drugs that can kind of fool a dopamine receptor into thinking it's received dopamine? Are there drugs that can inhibit the reuptake of dopamine so that the dopamine you are producing can have more efficacy? Uh, so those require, kind of a deeper dive into biochemistry. We're not going there tonight. Um, I think my colleague in the nursing faculty who does kind of the pharmacology of Parkinson's will be able to help us through that maze. But for now, I want to at least help you understand the difference between the blue spaghetti and the red spaghetti. So, so this is a single cell, single axon, bathing the input structure of your basal ganglia, the thing that goes wrong in Parkinson's, right? It's bathing that putamen at 350,000 places, but those places are not all the same. There's a red part of your putamen and a blue part of your putamen. The red part is receiving input from the motor cortex, it's kind of it's making decisions on what motions to carry forward, right? You've made a voluntary choice in your cortex, whether it's completely voluntary or not. I mean, you're reaching for the milk. Hopefully, that's we've done that so many times, or a beer, or whatever your beverage of choice. But there's a lot of competing motors, on your way to reaching into the refrigerator. Um, and so, for sure, there's going to be motor cortex, which we talked about before in previous talks. Your motor cortex should be involving the basal ganglia and choosing the right motor program. So so that's largely I think maybe that's the blue. The red is getting input from a very different place in the brain, not your motor cortex, but from limbic structures. Limbic structures are important in memory, association, emotions, um, and I don't have to tell you guys. I mean, that kind of Parkinson's is a whole brain disease. It's not just you don't just have motor symptoms. Um, and the cognitive things are a little bit harder to tease out. And so I want tonight, hopefully, to begin the discussion, of the non-motor inputs coming into the basal ganglia. Um, they have those in some sense those non-motor inputs into the basal ganglia also have a privileged access to dopamine. Um, and maybe that's not so surprising, right? I mean, so to people outside the Parkinson's world, they just associate dopamine with some kind of emotional rush, right? I mean, basically, I mean, it's a kind of a good feeling in some sense. Dopamine is needed to regulate motor, but it's also there, as the chemical of enjoyment, or addiction, right? And so when things go wrong. And so tonight I'm kind of opening the door to emotion. Um, and again, that's forced upon us by this Japanese tracing of an individual axon in the coming out of the Substantia Nigra of a rat, and seeing that really is spending some part of its time playing with synapses coming from your motor patterns, and and also spending a lot of time, so I'd say the blue zone, on messing with synapses coming from your limbic structures. Okay. And so that takes me to this was preparation for biochemistry. We can skip that for now. But the bigger cartoon now that I want to kind of help us understand, um, and it's um, yeah, there's so many beautiful things to say about this cartoon. So in some sense, hopefully there's not too many acronyms. Um, so dopamine receptors, by and large, come in two flavors, D1 and D2. Not much imagination there. So D1 and D2, but they're involved in distinct pathways between the input to the basal ganglia and the output to the basal ganglia. D1 receptors are involved in direct inhibition from input to output. Okay. Uh, and again, we talked about before, so too much inhibition in the network leads to the brain's inability to choose a muscle pattern, a motor program. And so if it can't choose, what we believe it ends up doing is allowing the body to execute multiple motor patterns at once, and that leads to rigidity, to stiffness, to freezing of gait, to lack of muscle control, because the brain is sending mixed messages down the spinal cord. Okay, so that's kind of the direct inhibition pathway. Now there's an indirect pathway, which is also inhibitory, right? So basically, these neurons in the putamen, they inhibit the GPE neurons in the external part of the globus pallidus, which in turn inhibits the GPI. And again, so this is a little bit of a logic crunch, but some of us are getting there, right? And I think if I say the right words, we get there, right? So this is inhibitory and this is inhibitory, okay? But if I inhibit my inhibitor, or if somebody inhibits my inhibitor, then I am free. This is called disinhibition, right? If I inhibit my inhibitor, then I am free to behave in a way, that that may be liberating or a voluntary emotion. So this is the classical pathway. This is the classical pathway. And again, I don't want to go too fast, but that's the pathway that was here. Okay. So there's a direct pathway where the putamen inhibits, so where the input inhibits the output. And there's an indirect pathway where the putamen inhibits the GPE, which in turn inhibits GPI. So, but if I inhibit my inhibitor, then the GPI is more free to do what it wants and to not over-inhibit the thalamus and cortex. Okay. So that's the classical pathway coming from motor inputs. And what's new that I wanted to share with you guys, and it's new like in the past just few years, is this understanding that that in the other part of the putamen, so I know in the rat picture it was colored kind of red and blue. Now I'm covered red and green. So there are kind of two parts to the putamen, and the dopamine is kind of bathing all of them sequentially. So the other part of the putamen, the striosome, right? It also has D1 and D2 receptors. Okay. D1, they act directly on the cells that produce dopamine. Okay. And they act indirectly also on the cells that produce dopamine, right? And so these are pathways that that we can potentially trigger with outside stimuli, like perhaps the glove. We'll get to that. Or there are also pathways for biochemistry to have kind of direct access on how to get the few cells that you have remaining, right? So Parkinson's is progressive, right? We lose more and more of these dopamine producing cells, but can we get the ones? Can we excite the ones that are down there to produce more dopamine? Right? So by understanding what is it that drives the striosome? The green part of your putamen is driven by motor inputs, which we spent a lot of time on in the past. The red part is driven by limbic structures, which is the subject that I'm hoping to prepare us all to enter into a discussion of. Okay, so we've talked about these motor pathways into the putamen as the input structure of the basal ganglia. The output structure then gets overly inhibited. Too much inhibition leads to poor motor program selection. Okay. And that too much inhibition comes from insufficient dopamine. But somehow dopamine is provided here, but these cells don't actually talk back to the dopamine producing cells. Where the other part of your putamen, the red part in this case, the red part does talk directly to the dopamine producing cells. So these are pathways that are extremely important if we're to understand how to remodel the brain. The brain is already making some right. The brain is accustomed to working in a kind of a dynamic environment. There are already kind of alterations made by the brain to deal sequentially, with less and less dopamine. Those are hard to understand because these are all really, really subcortical structures, and it's really hard to get our hands on exactly what's going on inside of these things. Okay, so so this is a cartoon. Well, also that hopefully the mathematician in you will love the fact, right? So basically, the basal ganglia has discovered a kind of part of it's going to act in a direct pathway and an indirect pathway on some structure. And then if you kind of sit back far enough and don't look at the letters, I mean, the diagram on the left is the same as the diagram on the right. It's just somehow, a different output structure over here. Okay. And so it's pretty cool that kind of once evolution discovers kind of parallel pathways of inhibition and disinhibition, it can then use that motif and other functions. So that same motif of indirect of direct and indirect parallel pathways from motor back to thalamus, right, is a very same logical structure as input structure putamen from limbic structures into the dopamine producing cells. So, and again, this is quite recent understanding, right? Because again, these are difficult things to tease out. but as a student, right? So it's beautiful when you learn like what are the building blocks of the brain? What are there any patterns, or is it all just chaos? Like a billion synapses in a cubic millimeter. My God, how can you get your head around that? So are there, there's excitation, there's inhibition, and there's disinhibition. And the brain somehow can regulate itself, can be more kind of robust if it does inhibition and disinhibition in parallel. As long as there's kind of a governor around, and the governor is dopamine. And without the governor, I don't need to tell you that things are not as planned. So I want to just I'm so happy with this beautiful picture. It's not mine, of course, but I want to show it to you in even in a slightly different fashion. To also just kind of see that these are parallel pathways from there are limbic structures, which we'll get to tonight, and then there are sensory motor structures. So before tonight, we spent all of our time on this pathway, right? So the body wants to move. The sensory motor cortex talks to the basal ganglia. The input to the basal ganglia is the striatum. I'm also using putamen. The putamen is a piece of the striatum, so I don't mean to be too fussy. Okay, so sensory motor input comes from cortex deep into this midbrain structure called the basal ganglia. It has two types of dopamine receptors, and they modulate different pathways: a direct pathway and an indirect pathway. Um, Yeah. And so that's the classical one. And this is the more recent one that's being studied, right? In this more recent one, limbic structures are coming also into the striatum, but the striatum is made up of these islands. Strisosomes are these kind of islands in the striatum. These also express the same dopamine receptors. Everybody's being bathed in dopamine. Um, there is a direct and an indirect pathway. Okay. This is the Substantia Nigra pars reticulata. This is not a place where dopamine is made. Dopamine is made here. Okay. So so as folks, well, everyone in the world just wants more dopamine, but especially you guys want more dopamine. And so how do we, so where do we intervene? I mean, do we intervene here or here or here or here? And so I am a student of kind of non-invasive measures for Parkinson's, so I'm not going to be intervening down here deep in your brains. I wonder, are there ways um, to speak to your limbic structures to give more excitation down here and less inhibition? Okay, so I want to talk about what are those limbic structures, right? Um, And and luckily for us, there's just two. We will just maybe barely touch on one tonight. But the two limbic structures that excite the striasomes, and again, to excite the striasomes is to begin the process of changing the amount of dopamine in your basal ganglia. Okay. So I want to talk about the insula or insular cortex and the cingulate cortex. Um, and maybe some word about limbic. I'm not sure if you guys are, it's an old Latin word for border. These were structures that were not immediately discovered in the brain. They're kind of on the border, kind of between regions, and it's taken quite some time to really tease out what are they up to, um, and and since they're kind of on the border and not directly implicated in kind of individual kind of sensory motor behaviors, it turns out that they're like involved in everything. That many people that are on the hunt for the seat of consciousness, for example, or the seat of the self, spend a lot of time in the insula and cingulate cortex because they're deeply implicated in the quest for understanding consciousness. Okay. But again, we're going after the brain for a purpose. I think we'd all like to understand consciousness better. So maybe I hope I might just be exposing you to some regions of the brain that are worthy of deeper study. Okay. Um, but what I really want to focus in on is the insula. So that's the green thing here. Can you guys see this kind of coronal slice through the brain? Coronal is like, cutting right through that center section. And there is a lobe of cortex, right, that kind of folds and it folds over its buddy as it goes down below. And just in this in-between folds, right, which you can't see unless you kind of pull those two folds apart on the brain. And there are plenty of pictures you can find online of doctors doing that. But this is just kind of the cartoon. This green thing is your insula, right? It's a piece of insula or insular cortex. And like I said, it's been implicated, in like so many possible, manifestations of what we are. And so I chose one quote from a recent paper I've been reading. So the insula plays insula for kind of insulated, it's concealed. It's a good name for it. Also kind of like an island. It plays an outsized role, outsized in the sense that it's not a very big structure. But like so much happens there. Bowel distension, orgasm, cigarette craving, maternal love. I'm going to get to maternal love. That's the one I highlighted. Decision making and sudden insight. And so, that that's a pretty big spectrum of stuff. I mean, we've been doing easy stuff, like reaching for the milk and things like that, and kind of motor programs. And we haven't really brought in kind of the more emotional side, the more cognitive side of the brain. So this is giving us a chance to talk about those things. Um, but I want to begin like what we've done before with just a little tour of the neuroanatomy. What does the insula look like, and how is it connected to other structures? Okay, and so how does information come into the insula? How does it then share this information with basal ganglia? And then once it's there, we already know what happens, right? From the basal ganglia, it makes its way to the cells that produce dopamine. Okay, so the insula is an input structure onto the cells to make dopamine. So it's worthy of our study. This little green worm on both sides. Okay, um, and so let me just begin with kind of the big brushstrokes, you guys. And so um, I'm not sure I can kind of go even any bigger than that thing. So um, I'm already kind of 200%. So it's a little bit small. But for what I want to talk about, this is pretty much the only thing we need to understand. And so the there is the anterior insula is the one closer to the front of your head, and the posterior insula is the one towards the back of your head. And then there's a medial insula that's kind of in between the anterior and the posterior. So that's the structure we're looking at. And you guys already know some of these other structures. So there's your thalamus, that relay station, between cortex and motion and sense. And this is the striatum. That's the input structure to the basal ganglia. And so what we see is the insula getting information, anything ending in a C here is cortex, right? So cortex talking to cortex, talking to cortex, talking to the insula, talking to the thalamus. And we see this kind of bidirectional communication between the insula and the striatum. The striatum for us is the input structure of the basal ganglia, the place that goes bad during Parkinson's. And then what about emotion? I talked about, maternal love and orgasm and all this kind of stuff. Um, the amygdala is down below. The amygdala is it's one of the emotional centers of the brain. When people think about limbic, they typically think about the amygdala and the hippocampus. Amygdala is this source of emotion. The hippocampus is the place where memories are stored. And they're totally related because we only store memories that are emotionally salient to us. We don't have the bandwidth to do a whole lot of storage. So only when something is emotionally salient, we grab it, we need it, does the hippocampus help us keep track of it. So those two structures are at play in kind of in the limbic brain. And which again is there's no such thing as the limbic brain, but just as far as separating it from the sensory motor brain. So there's the amygdala, the thalamus. So a few new brain structures for us that we haven't really thought about before, but you see they're parts of the pathways for the striatum. Okay. Um, and if we look closely enough, external stimuli and internal stimuli, the insula is also keeping a map of not kind of where we are in space, that's kind of proprioception, but interoception is, who are we? Keeping a map of our self through time. Okay. Um, by actually keeping track of things like heartbeat and respiration, but also external stimuli, what's coming through the nose, the ear, the tongue, but also what's coming through the fingertip. So this may give me a chance to kind of loop back to our some of our early times together by the glove, right? What can it do to the insula? So let me Uh, these are lovely also possibilities. If you spend any time on kind of part B of this picture, you're going to see that the insula seems to be implicated in so much of who we think we are. Okay. But I want to kind of focus in on pathways, understanding how things move between the insula and the striatum. And like I mentioned before, those pathways are pretty difficult to tease out. It's not ethical to tease them out in a living human. Um, we could argue whether it's ethical to do it in a rat, but it's been done in the rat. And so let's just take a look at what we've learned from that. So this is the rat brain. Um, and I guess I could go, I don't need to, I can always just talk through this, right? This is the rat brain. The big red thing is the insula. There's your thalamus. Um, cool olfactory bulb, sensory cortex. Um, yeah, so it's not even really easy to see, kind of where the basal ganglia is in that picture. But there's pathways from insula kind of all over the rat brain. Okay. And what I really wanted to show you in these pictures, um, I guess I maybe break it out a little bit easier here. Yeah. So in the front part of the insula, it talks a lot to the striatum. In the middle part, it talks a lot to the striatum. And in the posterior part, it talks a lot to the striatum. So these are kind of big red arrows. It's the biggest for the kind of the front part of the insula. And so what we're seeing is that if you look at the outputs to the insula, who's getting most of the bandwidth of the insula? Well, most of the outputs are going to the striatum. And then also to the amygdala and the thalamus. The thalamus talks to everybody as the relay station, right? But not necessarily the integrator of information. And so one of the biggest outputs of the striatum, sorry, of the insula is the striatum. Lots of stuff is coming. So then you wonder, well, where does the insula itself get its information? So what are the biggest inputs into the insula? And those biggest inputs are the amygdala largely and the thalamus. Okay. So amygdala means this emotional center, and emotions are deeply ambiguous, right? And so like half these inputs are excitatory, and the other half of them are inhibitory. As opposed to as we look to the insula talking to the striatum, it's all excitatory. Okay, it's all excitatory. So it's a fairly complicated picture, but just look at, find the biggest bars. Okay. The insula is sending the bulk of its output to the basal ganglia, the place we're most interested in. And it's doing that into circuits that are talking directly to the cells that produce dopamine. Okay. And so, yeah. And so I want to just leave you with a brief, kind of question, for me, a very pregnant question. Um, how do we how do we target the insula to be a better stimulator of dopamine in the basal ganglia? We see the pathways that are there. I know they're just there in rat. They're there also in human, but to less specificity than the bar graphs that I showed you. And so I showed you all those possible roles that the insula is playing, but there's one that's kind of closest to, we talked at length in a couple of sessions on proprioception, how the body knows where it is. And interoception is more than just kind of where you are physically, right? It's where are you kind of biochemically? Where are you emotionally? and even kind of, autonomic things, like where are you as far as, breathing and heart beating and like keeping track of those things. Visceral processing, right, is keeping track of of kind of sensory organs, that are not on the skin, right? So keeping track of how your digestion is working and the like. Um, and pain perception, a whole other thing we haven't really talked about, which is implicated by the insula. But I did want, again, just this is probably definitely a reach, but it's just I'm kind of I feel like sometimes I'm pushing guys over a cliff to try to gather all this kind of neuroscience data, and and we're all basically researchers on how the glove works. And so it's to me, and again, maybe it's just some kind of crazy coincidence, but the insula is implicated in this beautiful subject of effective touch. Um, and I have I don't have any kind of I don't believe. Well, I shouldn't say what I believe. We should just watch this. So, is anybody here kind of familiar with this term effective touch? Do you
No,
guys know this phrase?
first time I've heard of it.
Gotcha, gotcha. Yeah. So I'm gonna play a brief video for you guys of someone who I think really captures the power of this. It's super emotional, right? It is coming right out of the amygdala. Um, but again, we want to trigger the insula to deliver well dopamine for sure every place, but especially back to the basal ganglia. And this is happening in effective touch. Effective touch is again, it's definitely it's touch, right? Like what you're receiving through the glove, but it's touching a place, not your fingertips, and it's touching the place at a frequency which is not also coming from the glove. So I have this fantasy that it may be coming from other parts of the glove. So maybe the vibrations through your wrist, maybe the soft vibration of the cloth. So, in particular, effective touch only happens through your hairy skin, right? It doesn't happen on the palm of your hands. Most of us, I believe, don't have much hair left on the palm of our hands by this 21st century. But effective touch happens on the hairy part of the skin. So, I'm gonna. Oh yeah, I really want to make sure that sound goes through as well. So on my on my share, um, I really want to make sure that you guys can hear. Um, audio settings. I think there was a place. Um, share sound. Okay. Mm. So I'm gonna. It's just a brief video. Um, I find this woman quite
You
powerful.
have a literal switch in your skin that is pre-programmed to make you
Can you guys hear her?
Yes.
Okay. Okay. Um, and am I covering anything up with my? So
feel safe. Just unfortunately, you have to get somebody else to flip it. Inside your skin, there are these neurons, these little cells that will fire when they sense a particular type of touch, right? So some of them will go off when they feel pain, some when they feel hot or cold, some when they feel pressure. But in 1999, scientists made this really weird discovery. So they're like eavesdropping on some of these neurons using little electrodes, and they found one neuron in there that like basically ignores everything. Don't fire for any of that other stuff. Will only go off if you trigger it. The need like more.
oops, sorry.
You have a literal switch.
Sorry, I was just.
Four-legged forest clouds, the cowboy angel rides.
I'm hearing some Bob Dylan, I
This
think.
candle lit into the sun, though its
You have a literal
is
switch
waxed
in your
and
skin that is
bright.
pre-programmed to make you feel safe. Just
All except
Do you guys know where the
trees of Eden.
I don't think of any other sound going on. Sorry about that. Are we
With folded arms.
It's Julie.
it's Julie? Gotcha. I don't have a
I
big
think
enough.
I got it.
You got it?
It went off, right?
Okay.
Sorry.
Yeah. Yeah. Gotcha. Um,
Spontaneous Bob is always good.
yes, yes, yes. It's another way of saying, Steve, get on with this. Get on with this. Yes. Okay,
Sorry.
so I'm gonna go back to the beginning because she speaks pretty quickly, right? And again, the link is there on the notes. You guys can watch this. For me, it's a tearjerker. I mean, motherly love. What can go?
You have a literal switch in your skin that is pre-programmed to make you feel safe. Just unfortunately, you have to get somebody else to flip it. Inside your skin, there are these neurons, these little cells that will fire when they sense a particular type of touch, right? So some of them will go off when they feel pain, some when they feel hot or cold, some when they feel pressure. But in 1999, scientists made this really weird discovery. So they're like eavesdropping on some of these neurons using little electrodes, and they found one neuron in there that like basically ignores everything, won't fire for any of that other stuff, will only go off if you trigger it with a really soft little gentle stroke. Okay, that's the only way you can get it to go off. So they decided to call these C-tactile afferents. I mean, we know for decades that there's these types of things that you find in animals. Like this is the reason why when you kind of give a dog or a cat like a little scratch behind the ears and they just like lean into it and love it. This is literally what's going on, right? It's their version of these neurons that are firing and being like, that's delicious. But in humans, they're a bit stranger. Like there's some weird stuff going on. For example, like there's this very, very narrow frequency in which these things will fire. If you go too fast, it's not interested. If you go too slow, also not interested at all. It has to be this sweet spot in the middle of about 3 centimeters per second that will make these things go off. We also know that the temperature really matters of the thing that's doing the stroking, right? So if it's too hot, don't care. Too cold, not interested. The sweet spot, again, is about 32 degrees centigrade, which is around about the temperature of somebody else's hand. Now, okay, if you're somebody like most of us who enjoy the feeling of like soft caresses, then this is not going to be that much of a surprise. But the question is like, why? Evolution generally is about being as lazy as you can possibly be without dying, right? Why would evolution bother putting something like this, putting as though it's got agency, why would you get something like this as a result of evolution? And then, just a couple of years ago, this group of scientists led by Ilona Croy had this genius idea. They took a group of mothers and they put them in a room holding their babies, and they didn't tell them what they were doing. They just sat back and observed them. And every single one of the mothers started stroking their babies. They weren't just stroking their babies randomly. Every one of them was stroking their babies at exactly the frequency that these neurons fire at, right? This is like no training, no kind of calibration. This is just like maternal instinct that knows the exact rhythm to make their baby feel good. And we now know that this neuron firing, right, is linked with lowering the baby's heart rate, reducing the baby's stress. And if you do this for preterm babies, you will literally reduce the amount of time that they spend in hospital. How gorgeous is that? That there is this like built-in biological lullaby that we are all pre-programmed to know the rhythm to even before we're born. What an added win that it just so happens to work for adults too. You have a literal
Yeah, so that's effective touch, and those C-tactile afferents are in your hairy skin, and they're processed in the insula. And just to kind of review, the insula is that has the place in the limbic structure that shares with motor cortex inputs into your basal ganglia. But unlike the motor inputs, the inputs from the insula have the impact of actually allowing you to create more dopamine. And again, it's not surprising. I don't know, did you guys get a dopamine rush when you saw these little babies or even the cat, kind of bowing its head under the touch? So there is something about the glove is about touch. The frequencies have been chosen based upon mechanoreceptors in your fingertips, not upon what's happening on the top of your hand. Um, and so, so again, it's a little bit of a reach, but for me as a teacher, just tie things back to where touch might be happening. We are trying to make kind of more stable, more robust glove, for folks. But maybe in kind of over designing stability and robustness, we might be losing that kind of soft touch on the top of your hand that you guys are getting from these designer fabrics that we've been producing in Espanola. So So that's where I wanted to leave us for tonight. But I would love to hear your thoughts on where we've been, what we're doing, and your thoughts that especially kind of new folks coming that want to hear from. So Lauren and Nico and Julie are joining us for some of the first times, where Laura McClure and David and Roger have been wearing the glove for a longer period. So I'd love for you guys, and Jennifer as well. Sorry, is new to the glove world. Um, Oldest veteran, I shouldn't say oldest, not really the right word. So our most senior veteran, wearing the glove the longest, um, is is Frank Grubbs, who's on the line with his wife Pam. So Jennifer, please.
Yeah. Have the people who have been wearing the glove for a while, have you had improvements in your symptoms?
I want to say that um, I've been wearing the glove for probably 6 months, not your gloves, Steve. In fact, I just received your gloves and they're being shipped to me. I should get them next week. Um, I have some gloves that follow the task protocol, and the thing about the gloves, it seems to me, and I wanted to mention this earlier, is that if you're feeling very tremory and you put the gloves on, it's not gonna, from my experience, it doesn't stop it. So it's relaxing, it feels good, but I think the impact of the glove is from repeated use consistently. And so I think it's reorienting and reconditioning, reprogramming part of your brain, and you don't have immediate effects from it. That's my experience. So I'd be interested on what other people think about that. Do you feel immediate relief from it, or is it something that it takes a while to start to notice a difference, and you can even not use it for a couple days and it's okay. But then if it's longer than that, you have to kind of start all over again. You've lost some ground, is what my experience is. But
Thank
again,
you.
I'm not getting an instant response.
I would agree with that completely. That is very well stated. I also, it's more of a cumulative effect than an instant response for me.
And it's
Uh,
different from deep brain stimulation in that regard because deep brain stimulation is attempting to correct it at the moment.
Mhm.
And so it seems like what you're trying to work on, Steve, is a way to take some sort of sensory response and have it change the vibrational pattern of the glove to try to spontaneously or instantaneously affect a person's dopamine or
Yeah. I guess, but not necessarily instantaneously, though. I mean, I guess that there still is, it's still indirect, right? We can't go down into those structures, but we can through plasticity kind of work on somehow rewiring of circuits. But that plasticity is probably sensitive. Every Parkinsonian patient is different, right? And the sweet spot for that timing sequence through the fingertips should likely be better tuned. And so some people do respond acutely. Like in the Tassa's trial, like 3 out of the 6 people did respond within the first weekend. Frank Grubbs responded pretty quickly.
Right away. Yeah.
Yeah, he wanted to share it, but
Frank?
Frank, can you speak?
Yeah. Right away, I was looser walking. My gait was better. It was definitely noticeable.
Yeah, same day.
Yes.
Yeah, for Frank, we both noticed a difference right away.
so
Has
And I
that continued?
My arms swung really easily.
So if you're off and you put the gloves on, do you instantly come on or
Well,
I feel like if I'm off, like shaky, and I use the glove in about 20 minutes, I calm down. So it's pretty it works for me where it just calms my body down.
How long do you wear it for?
4 hours a day, 2 hours in the morning, and then I do it at night because I don't want to wear my gloves when I'm on. I want to get things done, and then when I start feeling shaky, that's when I put them on.
Has anybody reduced medicine?
I have.
Really? Oh, that's
That's
great.
that's with my neurologist, and she wanted me off of Rapidinol, so I don't take it anymore.
Oh yeah, I did not like that drug.
Yeah, and it took me a while to titrate getting off of it, but now.
Yeah, that was kind of rough.
Mm-hmm. And then also with the carbidopa, Conwa was telling me I was overstimulated, so I take a little bit less, like I take 3/4 of a pill instead of a whole pill, because I've been getting bad reaction to the, like twitching.
Yeah.
So I'm finding that I need a little less. But
Oh, that's wonderful.
yeah, but like what Nico was saying, everyone's different. We experience it so differently.
Yeah.
So what works for me may not work for somebody else. It's
How
kind
long were you using the gloves when you noticed a difference and you were able to eliminate a little medicine?
Probably 3 months.
Okay.
Now I'm up to like 5.5 months. But I just
Thank
find
you.
I look forward to using them. It calms me down.
Yeah. The vibrations relax me.
hmm But I think the other thing is when I'm very trembly, I do deep breathing with the gloves. I think the breathing really helps calm my system down too.
I actually use mine to stretch and exercise when I'm wearing them, and it
hmm
feels really good to stretch your arms out all the way and
Right,
Yeah.
make some motion.
Yeah, that's what I do first thing in the morning. When I wake up, I put them on and I get up and I stretch and I do my stretch routine and with them on.
right. I break it up. Like I sit for some, stretch for some, walk for some, sing for some.
Yeah.
2 hours is a long time.
Yeah.
Mhm.
Sometimes those 2 hour sessions do seem like a long time. I'm
Right.
thinking, like, to go to write something down. It's like, oh, nope, can't do that.
So I break it up, but and you know, I was told morning, afternoon, but I do late morning and early evening.
Okay.
That works with my schedule, I guess.
I've had good results with sleep.
Me too.
Before using the gloves, I would wake up anytime 3, 4, 5 o'clock in the morning and not be able to go back to sleep. And then throughout the day, towards the end of the day, I would be exhausted. And wearing the gloves, I'm sleeping all the way through the night. The other morning, I woke up at 9 o'clock. I couldn't believe I slept that long.
That's exciting.
Yes, it's
That's
wonderful.
fantastic.
Yeah.
Yep, that was the first thing I noticed was being able to sleep better.
Hmm.
I don't know if it's because I feel like listening to you that strengths maybe further progressed, but when he like has done the gloves for a couple hours at night, sometimes there's a bigger reaction than others, but often he'll be perky and he'll go look at me and he'll walk real fast and it's like, okay, well now you need to vacuum. It's like let's go somewhere, but it's late. But the difference same day even is dramatic for him. And it does. We have noticed that for whoever asked earlier, if he does go a few days without the books once when they were being repaired, there was definite regression, whatever, over a several day period. But, so a lot of the help lasts, but right afterwards for him, it's pretty dramatic in his walking and speaking.
I've had that happen occasionally where when I was finished with the gloves, I was normal. It was totally amazing.
Yeah.
It was such a joy. And then it went away.
Right. Yeah. I wish it would last.
Yeah.
It's exciting when it happens.
Yeah.
Yeah, so that's so tantalizing. That's just it's so heartbreaking, right? To be able to deliver and then have something. I mean, it's just like levodopa, right? I mean, the brain habituates, and so my fantasy is, that by proper monitoring, we can tweak, from afar, that we can tweak the glove so that it keeps up, with you as a person and not just doesn't just give you that one window, of kind of symptom-free, life.
But
I have
it's
a question. When I wear the glove, I feel like I have a vibration that starts on my right hand on the pinky and goes to the pointer finger and then across to my left hand. And so I have stimulation on the first couple of fingers, then the middle fingers, and then the end of my left hand. Does anybody have that kind of experience?
it travels.
Yeah,
So
it travels
right. Yeah.
from my right pinky to my left pinky across my two hands.
Well, and then sometimes I listen to it.
Mm-hmm.
And you can hear it traveling.
Uh-huh.
Mm.
Well, there's a lot of variability in the programming, and there's many different ways to deliver this therapy. And for example, the task glove has a jitter in it.
Oh,
Steve, you and I spoke about
Yeah,
that.
yeah, yeah, yeah,
And they also have them coordinated. So the two gloves are connected. So they're working at the same time. It's called a coordinated reset, is what Task calls it.
yeah.
neat.
Yeah, so the gloves that you guys are getting from Espanola, those are not coordinated. They're each you've got a separate computer on each wrist. It would be if we use the app, it would be possible to synchronize them. TAS's research didn't really. The research said in the small clinical trial that kind of the more jitter, the better. Kind of the more kind of randomness.
Exactly, yeah.
But from my understanding, the jury's still kind of out on whether the coordination of the two hands was necessary. So Lauren, it's fascinating, Lauren, that you really feel a wave going across the fingers because the finger they're not really they're not kind of stimulated sequentially across. But I think, I mean, at least when I wear them, I really my brain just immediately like hunts for the pattern, right? And it's not there. But maybe if you relax enough, kind of step back from trying to actually find the sequence, your brain, just basically fills in the fact that a wave is traveling, from one hand to the next. Um, I'm not programming jitter into this because TOS was using heavily engineered motors that cost hundreds of dollars, and I'm using a $1 motor off of Amazon, which has its own kind of built-in jitter just because of a simple piece of hardware. So that's not an excuse. But it's a way to kind of keep it cheap. I shouldn't say cheap, to keep it inexpensive or kind of available to folks who just want to experiment with it. And so we can use kind of jittery hardware or jittery software. So I'm using jittery hardware right for the moment, and with some results as we're hearing from folks tonight. So it's not like it's not just a cheaper version of TOS. We are getting some jitter.
So
If
should
that's
is it real what I'm perceiving that there's a move a traveling of the stimulation?
Yeah, so that's a good. I want to be respectful, Lauren. So it's not as far as the engineer or the designer, it's not real. There's not a sequential kind of it's not moving across the hand from one to the next. So I believe though that's from my experience that's the brain kind of filling in the pattern in between, right? And so when we're faced with novel stimuli, we typically match it onto something that we already know, right? And so as Laura talked about, kind of the music traveling across, your body, your vibrations traveling across, it makes sense that your brain would fill that in. If I understand TOS correctly, I think that basically these patterns, since they're not predictable, they're tricking the brain into receiving stimuli that it doesn't know what to do with. And so it doesn't know what to do with it. So it basically breaks connections in the brain that would actually be overstimulated by the lack of dopamine. So that was the subject of a couple previous talks together. It's a little bit complicated, but Tass argues, at least kind of numerically, that by delivering these stimuli that don't really lead to kind of effective movement or any next step, or just kind of buzzing a finger and stepping back, that's actually reducing synaptic weights between cells and cortex that have gotten kind of too close together because of the lack of dopamine.
And that's why there's a pause.
Yeah, yeah.
So it's not constant stimulation. And actually, the experience that we've had in developing this, the earlier version that I had, um, it wasn't random enough. And um, if you think about it, even if you had 1,000 different variations, it's still not random. And you may not feel it on a conscious level, but your brain is receiving it and using it. And so
Processing.
um, I found that really interesting. So as I was saying, the earlier version didn't have much of a jitter, if you will, or the pattern wasn't that significant. There was maybe 7 or 8 different patterns, and it became annoying.
Hmm. Yeah, so that's a word I've not heard people use for the glove that we're making an Espanola, right? They feel there is some kind of musical pattern. It can be kind of joyful or relaxing. People are able to go. Some people wear it to bed and are able to fall asleep with it. So, but is that so? I didn't mean to burst any bubble, Lauren, by saying so. We're not delivering that wave, but it's not surprising, that the brain is actually, visualizing or feeling that as a wave. Um,
I changed them yesterday to I had conceptually a right and a left because of
uh-huh.
the pattern. Uh,
Yeah,
and I developed that perception because I put them on different hands. I exchanged the hands
gotcha.
and I had a whole different experience with it.
Hmm.
So we shall see. It's only
That's
a couple of
fascinating.
days I've had them.
Yeah, 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 yeah. These are early days for all of us, to tell the truth. I
Yeah.
mean,
But I'll tell you, having a good night's sleep, there's nothing like it. If it only delivers that, I'm
gotcha.
delirious.
Yeah.
I don't know if anybody's heard of the Beach Band. There's a guy named Carl Beach who
Hmm.
is in the UK,
Yeah,
and he created basically a watch that has a tactor, a vibrator in it, not four on the fingers, just a single one. And he's claimed that and he claims his results are cumulative. And over a year and a half, he's basically off all meds and he feels like he's completely cured. And he encourages the regular use of this product for hours and hours a day, as long as you can use it. And obviously, it's much easier to use if it's on your wrist than your fingers because
yeah,
having it on your fingers
Yeah.
limits. But there's, as you said, Steve, there's many different ways to accomplish this, and we're in the very early stages. But it's really interesting to see what he's done in his experience with a different type of stimulation that's not even on the fingertips; it's on your
yeah.
wrist.
I
Well,
actually have one, a beach band. This is Marcia talking. And I received it probably 2 months ago. I had a friend in England get it for me. And actually, today they just became available in the United States.
Hmm.
Um, it's interesting. It's a different experience. I find it sometimes annoying to wear it on my wrist, and I wear it on my ankle instead. Um, the gloves have more of a soothing effect for me. Now, I am tremor dominant, and really that's pretty close to my only symptom. I don't have problems with rigidity or balance or slowness. But I have a lot of anxiety, which I've had all my life, and a prominent tremor. So what I did was I try not to mix the beach band with the gloves because I think it's going to muddy the waters. And now I'm wearing the gloves more consistently over the beach band. And I'm not on any medication. So I'm trying to just see what I can do with this alone. I don't know if that's helpful for anybody, but I offer it up.
Thanks, Marcia.
You're welcome.
I have to sign off. I'm sorry.
No, this is it's 8:15. I didn't mean to keep you, but I just want to give people a chance to share and get to know each other. This has been a great discussion. So yeah, I'm gonna turn off the recorder. I'll stick around if people want to have any questions for me. Um, and I wish each of you kind of a happy Thanksgiving holiday. And I'll see you back here in 2 weeks. I'll
Thank
post a
you.
recording of this soon.
Thank you.
Thank you,
Thanks,
Thank
thanks,
Steve.
you.
Jennifer.
Great to meet you all and hear your experiences.
You as
Same here.
well.
Yeah, vice versa.